Hey everybody, welcome back to yet another journal club. Man, it seems like we just had one a few weeks ago, and we did 2 weeks ago, right? We had now two for December, getting these things, banging them out for the holidays. So early Christmas for everybody, the journal club. So welcome everybody to Lifespan.io journal club related to aging. As always, I don't think we discuss papers that are not on the topic of aging or longevity or rejuvenation therapies. So here we are. I'm Dr. Oliver Medvedik. And what do we have in store today? Let's share the screen. There it is. A drug-like molecule engages nuclear hormone receptor DAF-12 FXR to regulate mitophagy or mitophagy, never know where to put the stress on the pronunciation, and extend lifespan. So this paper deals with actually a couple of different organisms. It deals with mammalian cells from mice. I believe they might have human cells. I think it's primarily mouse cells. And lifespan extension in C. elegans. So this is a paper from the Buck Institute, Chamoli et al. And this came out fairly recently, I believe just last month. So it's a Nature Aging paper. And this kind of belongs in a class of papers that we've covered before where they've looked at small molecules that extend lifespan that are derived from plant compounds, so things that we eat. One of the compounds that they're looking at here, the drug-like molecule, is actually a benzocoumarin, which is found in foods. So I believe it's this multi-benzene ringed molecule. And it tends to have beneficial effects across species. So we've looked at a number of papers where people have been isolating these molecules that have, beneficial effects kind of up and down the evolutionary ladder throughout species. So pretty much indicating that these molecules are targeting pathways that are fundamental to the aging process across many organisms. So therefore are probably very promising to work with. So I'm gonna stop sharing here, drink some coffee. Anybody have a chance to read the paper? I know there was kind of short notice since we just had one before this, and now we've got this one here.
Not I.
Anybody have their fill of benzocoumarins? Yeah, other than being present in a variety of foods and plants, I don't know which plants would be the most enriched in these coumarin-like molecules. Anybody here happen to know off the top of their head? No.
Find out for you, probably.
No, that's okay. Okay.
I'll get back to you.
You'll get back to me. Okay, excellent. All right. So, what is DAF-12 and what is FXR? So, DAF-12 is the ortholog of FXR, which is a nuclear hormone receptor. DAF is the ortholog in C. elegans, FXR in mammalian cells. And there's this whole class of these nuclear hormone receptors. They bind to small metabolites and switch on genes, and some of the small molecules they bind to are hormones. So let me share my screen here. This is just just grabbed this screenshot from somewhere. God knows where on the internet. I didn't make this from scratch. So here you have a hormone that enters the cell through the plasma membrane, binds to a cytosolic floating nuclear receptor. This targets it to the nucleus. They tend to either dimerize or interact with other partners, and they bind to their hormone response elements. And when they do that, so they have a DNA binding domain, they have a ligand binding domain. And when that happens, they can switch certain genes on. And in this paper here, they implicate this DAF-12 FXR being in this pathway in which the benzocoumarin I mentioned is operational. That being said, I'm going to stop sharing here, and we'll share the paper in a moment.
Quick question. Hormones are proteins themselves, right?
Well, yes and no. Sorry, I responded too quickly. So I believe for the nuclear hormone receptor hormones that are binding, they're not proteins. They're small molecules such as estrogen, testosterone. So those are cholesterol based. You can't have hormones that are peptides. You can definitely have hormones but those I believe bind to external receptors on the plasma membrane. I don't believe that the ligands are proteins for nuclear hormone receptors.
Okay.
Yeah, but hormones in general can also be proteins. So hormone by definition is basically something that is released in the blood from one organ, from an endocrine gland, and travels the distance in the bloodstream to a distant target site, distant cellular site along the bloodstream. So basically, long range acting molecule that switches genes on. Okay. So the one caveat I'm going to mention here is I've read this paper a couple of times. I'm still not exactly sure how DAF-12 FXR exactly functions in the pathway of the small molecule, because the image I just showed you there is that these molecules bind to nuclear hormone receptors, and then they bind to promoter elements, and then they switch on the genes. So that seems pretty straightforward, except they have got some assays in this paper which kind of seem to contradict that, and maybe I'm just reading them wrong. So you all can educate me on this. Got a comment here, and retinoin is a non-steroidal ligand for a nuclear receptor. Thyroid hormones too. Yeah, okay. Thyroid hormones too. Are thyroid hormones peptides, Sven? Thanks, Sven. Just want to take a break. Okay. So maybe there are. Yeah.
Maybe I can jump in by voice because I'm trying to chat. I write in the chat on my phone, and that takes a bit of time.
Okay. Sorry.
And I have gloves on in the lab, so that makes it even worse. Okay. So thyroid hormones are actually small molecules. They are one of the amino acids, I think, tyrosine, and then they are modified with some iodine to it.
Okay, so they're actually got kind of a modified amino acid there rather than a small protein.
Yeah. They are definitely a small lipophilic molecule.
Got it. Okay, so small molecules in general, so not something larger like a polypeptide. Okay, excellent. So let us go back to the actual paper. Okay, so what compound are they looking at? There's no picture of it, so you can Google how these classes of small molecules look like, these coumarins. So they did a drug screen, and they looked at a natural product library, and they discovered a benzocoumarin that they call mitophagy-inducing coumarin. I believe that's what they're calling it. A benzocoumarin compound. I don't know exactly if they mentioned which type of benzocoumarin compound they isolated, but they're calling it MIC, mitophagy-inducing coumarin, not to be confused with CMIC or many of the other MICs that we've been referring to here for papers, but this is their own here. And they note in this screen, which I'll scroll down to in a moment, that it enhances expression of this it enhances TFEB expression. So TFEB is a basic loop helix transcriptional factor, and it serves as a master regulator of genes involved in something called ALP. So a lot of acronyms floating around here. So what is ALP? Not to be confused with ALF. ALP is autophagy lysosomal pathway, right? So that's what that is. And so they're going to try to figure out in this paper the mechanism by which mitophagy-inducing coumarin basically promotes mitophagy, right? So mitophagy is essentially the turnover of mitochondria. So very similar to autophagy and other types of internal phages of the cell, where you essentially have anything that's problematic, anything that's misfolded, essentially get targeted to lysosomes and broken down and recycled. So this is happening at a larger scale with mitochondria, and that's the focus of this paper. So they note that this mitophagy-inducing coumarin, as the name indicates, induces mitophagy. So the question is, how exactly is it inducing mitophagy? And what does that mean with regards to the lifespan of C. elegans? Okay, so how do they find this molecule in the first place? So it's kind of interesting here. I mentioned that they work with several different species. So the actual screen that they used to identify this compound was using mammalian cells. So this was N27 rat dopaminergic neural cell lines. So these are dopamine responsive neurons. They were, I don't know why they picked these specifically. They mentioned that they were trying to look for something that targets, again, to have something that has translational relevance to mammals as well, because later they look at C. elegans. And so they're easily cultivated, so that's good. That's one reason they use it. But I'm not sure beyond that why particularly, other than being easily cultivated and cross-translational, they're using this exact cell line, but they use it. And the screen that they use is that they have this reporter gene. So they want to see what can turn on TFEB, right? So there's a TFEB promoter. So it's a promoter to turn on this this TFEB basic helix loop helix transcription factor, which basically regulates a whole bunch of downstream genes that are involved in mitophagy. So they wanted to see if there's any compounds that switch this on. So they want to see if expression levels of this TFEB go up in response to this promoter. So the promoter is driving a reporter gene, luciferase, I believe in this case. I don't know exactly how many compounds they screened, but a whole lot. And they basically tell us that their biggest hit, which gave the most fold expression, was this coumarin, benzocoumarin based compound. Oh, there it is. So there's a little picture of it right here. So multi-ranked compound. So this MIC compound boosts levels of TFEB. Well, actually, it interacts with the promoter. So so if you have this the system in these cells and you add in MIC, then levels of expression of the reporter go up. So somehow, we don't know yet how, somehow this compound is switching on the expression of TFEB. And in C, what you see here are basically downstream targets of TFEB. So there's additional genes that TFEB will switch on. So when they look at mRNA levels of these, they go up as well. If you increase concentrations of this benzocoumarin from 10 micromolar to 50, you basically have, looks like a linear increase as well. So that's basically some background. They then follow this up by having another reporter system where instead of TFEB, so there's another ortholog, so something that's similar in a different species to TFEB is in C. elegans. There's the protein HLH30, which basically is basically the orthologous to TFEB. So earlier I mentioned DAF-12 FXR, which is a nuclear hormone receptor that they're going to implicate in this basically this pathway to switch on TFEB. But right now they're switching to a different model organism and showing that if you add this benzocoumarin to C. elegans that has this reporter integrated within the worms, you also get higher levels of GFP intensity. And they also do a Western blot analysis of HLH30 itself within the worms, and that tends to go up with MIC concentrations, and also a whole bunch of downstream genes that are analogous to what we saw here that are switched on by HLH30, aka TFEB in mammalian cells. So that goes up as well. Any questions here. So you add a benzocoumarin and you get this transcription factor that is known to switch on things that are involved in mitophagy. It goes up. So later, so now they start to kind of go into, does all of this actually correlate to, correlate to improved mitophagy? So they do another assay here. This is something with a stain called lysotracker red, which I believe this stains active lysosomes. So let me just kind of orient myself to this figure here. Let's see, what's this figure 1H? HI. Yep, so it's a cytotropic dye that stains lysosomes. So if you get an increase in this red fluorescent signal, you basically have increased presence of acidic lysosomal compartments. So that goes up as well. So does anyone happen to have a copy of the paper downloaded that they can share? Mike is asking for the copy. I think it was sent on the email. There is a, it should be attached somewhere. I could share that with you. Like, do you need it? You need it right now? Let me see if I could.
It'd be nice to be able to follow along.
Uh-huh. Steve, do you have a copy you can zip over to Micah's way?
I do, but my keyboard's not working. So,
oh boy.
I can't actually do anything at all, but I dare not touch anything.
Okay,
all the keys are doing different.
So we just went through figure 1, so we're taking a little break here, folks. Let's play Q. That's me.
If you'd like to buy me a new keyboard.
Why don't you continue, Oliver, and then if Steve's keyboard ever works, then he can send it over to me. Okay. Otherwise, I'll just follow on your screen.
Okay, fair enough. So that was figure 1. Kind of breeze through that. So far so good. It seems pretty straightforward. You add this benzocoumarin in there. The control was DMSO. You get a lot of, the only thing I would mention here is kind of a caveat is whenever I see something like this, are there other genes being expressed as well? Because, okay, yes, you get mitophagy genes being turned on, and as a consequence, you have increased mitophagy. But what else is this benzocoumarin doing in the cell? Might be targeting a lot of other things as well. Albeit the focus here is on these mitophagy genes. Okay, so TFEB is implicated, HLH30 is implicated. It's a transcriptional factor. Add a benzocoumarin, you get higher expression levels, and things downstream happen, which, as you would expect, if the levels of this transcriptional factor, that's a master regulator, go up. Now, the next question they want to ask is, well, how is this transcriptional factor being switched on? And this is where they implicate the intermediary of this nuclear hormone receptor, which they implicate as being either directly or indirectly, I'm not quite sure about that, being regulated by the benzocoumarin, which then is switching on the TFEB. And this is where things get kind of a little confusing for me, because I showed that figure earlier that basically very detailed Microsoft Paint model of nuclear hormone receptors. So they do another screen here, trying to implicate or another, they set up another assay to implicate DAF-12 FXR. And the reason they initially started to look like what kind of triggered them to implicate DAF-12 FXR is that HLH30, the coding sequence for it, and also FXR, I believe, in mammalian cells, the promoter has these DAF-12 binding motifs. So these elements that either DAF-12 or or the orthologous nuclear hormone receptors in mammalian cells would bind to. And there's also, there's also DAF-12 binding sites in a lot of things that are, a lot of other things that are kind of implicated in mitophagy as well. So I mentioned in the earlier figure that that the transcriptional factor TFAB and HLH30 switched on genes downstream because it in of itself is a transcription factor. And those are genes implicated in mitophagy. So I'm not sure if these other genes also have additional, TFAB binding sites as well. But that being said, they have these DAF-12 putative binding sites. Okay, so so what did they do here? So they look at HLH30 messenger RNA expression. I think we already know this, that if you add in MYC, or is this they do a separate Jasper core. So identify this. So they did ChIP-seq data, verified 12 DAF-12 sites in the promoter. Okay, so we know that HLH30 messenger RNA expression level goes up, and a lot of these other genes that they look at later that also have DAF-12 binding sites also goes up. Now here's where things get a little bit confusing to me. They set up something called, and you'll see later kind of what I'm talking about. Maybe I'm kind of misinterpreting this a little bit. So they want to see exactly how this benzokumarin, which they call MYC, mitophagy-inducing kumarin, how it may possibly interact with DAF-12 or nuclear hormone receptors in general to, I'm assuming, switch on transcription, right? So they set up a one hybrid reporter assay. So just to back up a little bit, this was initially a yeast-based reporter assay that, when it was first published back in the 90s, it was called a yeast-2 hybrid system. And the way these systems generally work is that, just like with nuclear hormone receptors and lots of other things that switch on genes, the way that a lot of transcription factors work is that they have two parts to them. They have a DNA binding domain that binds to elements in a promoter, and then they have an activation domain. And what the activation domain does is it recruits or it binds to additional transcriptional factors, but more importantly, RNA polymerases, which then once they're at this location can start essentially transcribing the gene, right? So the way that these two hybrid systems work is that you have you take this gene that's a transcription factor in yeast that binds to genes that are involved in carbohydrate metabolism, so galactose, and this transcription factor is called GAL4, and it has a DNA binding domain and it has a transactivation domain, and you could split it into two parts. You can have one component have just the DNA binding domain, and one other component, by component I mean a protein, have the activation domain. And then what you can do is you can make a fusion protein of your favorite protein to one part, and then have a library of all sorts of proteins to the other part. And if those parts interact inside of the cell, they'll form a complex, and that activation domain will synthetically be brought back together with its partner DNA binding domain, and then you have transcription basically switch on. So it's essentially a way to figure out, a way to isolate things that interact inside of a cell in vitro, and identify partners. This one hybrid reporter assay basically does something very similar, but instead of having the activation domain floating around attached to a GAL4 activation domain and having an unknown protein, what they're essentially doing here is they attach this DAF-12 ligand binding domain. So DAF-12 also has two parts. It has a ligand binding domain. So it's a nuclear hormone receptor. So a ligand is the small molecule or something, a metabolite that binds to the DAF-12 or the nuclear hormone receptor. And that induces a conformational change and binding with a partner. And then its DNA binding domain will then attach to a promoter. Okay, so this is an artificial system here. So if the ligand, and I think that's something called dafachronic acid, which is the native ligand for DAF-12, binds here, it will undergo conformational change, and other factors, including RNA polymerases, will then be able to bind to this and switch on essentially expression of the reporter gene, in this case being luciferase. So no ligand, no binding, ligand binding, right? So, and they use this, I believe they use this in human embryonic kidney cells, 293 cells. I'm not sure why those cells in particular for this assay, but human mammalian cells. So what are we seeing here? So what they're showing here is if you add in this benzocoumarin that they initially identified in an earlier screen, which is mitophagy-inducing coumarin, that's what they're calling it, so we'll just call it MIC. It tends to bump off dafacroic acid from the ligand binding site. Okay, so you have now reduced levels of expression from this luciferase reporter. So that to me implies that either directly or indirectly, you have this benzocoumarin competing with the ligand at this location. So perhaps I was thinking, well, maybe that's how it works, is the benzocoumarin is a much better ligand than the dafacroic, than the dafachronic acid. But if that was the case, then you would expect higher reporter expression levels, because if the benzocoumarin bound, then it would perform the function of the ligand. So then perhaps I thought, well, then maybe the benzocoumarin is actually kicking the ligand off, and it's the unbound version that is switching on the reporter. But no, that's not the case, because this figure shows us that it's only if it's bound, then you actually get expression happening. So they go on with these further assays to basically show that and now they have a similar worm-based system. So they have a myo2 promoter. So this is using DAF-12 in worms. So a very similar reporter-based system, kind of a one hybrid-based reporters-based system, and also showing that that either MIC will turn this off, or they add this compound called DFA. And what is DFA? DFA is let me just pull this up here. It's a figure legend 2F Dafidine A. It's a specific inhibitor of the DAF9 CYP enzyme, which is essential for the final step of DAF-12 FXR ligand synthesis. So basically what this is doing is this inhibitor knocks down dafacroic acid expression level, synthesis levels in the cell. So you have less of the ligand, so you have less switching on of the gene, so less GFP expression. So I believe here they're basically looking at GFP also as a Western blot, and MIC is performing the same thing. So I'm going to stop sharing here. So it's clear to me that MIC is interfering with the binding of the native metabolite that binds to the nuclear hormone receptor here or the ortholog of the nuclear hormone receptor here, which is DAF-12 and C. elegans, using these assays. So that tells me that so this is where I got a little bit confused. So if DAF-12 is switching on TFEB, and TFEB is being switched on by DAF-12 that's ligand bound, then shouldn't the levels go down then? And I'm not sure. So clearly, there's a bit of a confusion here, and I've been looking at this, and I can't figure out what is the error in my logic here? If anybody else can spot that and let me know, because that's been really kind of getting under my skin. I can't figure out if it's a good thing. Well, yeah. So what I am sure of is that this benzokumarin is interacting with DAF-12 to some capacity, and it is seemingly disrupting the interaction of its native ligand. And that's supposedly is a good thing with regards to lifespan, because it's going to upregulate mitophagy downstream. Okay. Usually these things are pretty straightforward for me. Take a look at the figure. You're like, okay, ligand binds, things are on. Ligand doesn't bind, things are off. But unless there's some sort of kind of dual stuff happening here, I'm not too sure. Okay. So this is regardless. And again, kind of and DAF-12 is important for this, because they later mentioned in the paper that, and I think they do some further experiments where they have DAF-12 mutants or null mutants where they get rid of DAF-12, and this basically abrogates the functionality of MYC. So basically these mitophagy induction, you don't see that anymore when you don't have DAF-12/FXR in the pathway. So it is being regulated by MYC. I'm just not really sure, based on the screens I saw, whether it's positive regulation or negative regulation, which is a little frustrating to me. Okay, so what else do they do? So now they're again focusing on C. elegans. So bulk of this paper, the screens were in. It's interesting. The screens were in mammalian cells. Either was it rat or mouse neuronal cells, and some of the other assays were in mammalian cells. But a lot of this work here is in C. elegans. So it's nice to see a molecule like this have kind of very conserved cross-reactivity across many different species, evolutionarily speaking, suggesting that it's targeting some really fundamental mechanisms and some very conserved pathways. Okay, back to the paper. So what do they do here? So now they basically take a look and see what is happening with mitochondria when they're adding this compound. And they basically, in figure 3, it's all about MYC-induced mitophagy enhancing mitochondrial health. How do they do that? So they have GFP tagged mitochondria in intestines in these worms. And they're adding in MYC and then seeing a whole bunch of different things. And what are they seeing? They're seeing mitochondrial size go down, which they indicate is kind of indicative of mitochondrial turnover. They see expression of this gene DRP1, which is basically, I don't know exactly what DRP1 does, but essentially it's involved in mitochondrial fission. So mitochondria replicate, they undergo fission, so they kind of autonomously do that. So you have mitochondria dividing, you have smaller mitochondria, and then they add these two toxins, FCCP and rotenone, and I believe rotenone is an herbicide, so that you can, so
it's an extreme powerful herbicide, quite dangerous actually. People that lobby against Roundup, I'm sure we've all heard about that. They tested it. Yeah, they tested it against a number of these organic pesticides, herbicides, and Roundup is actually a lot safer or much less potent. Yeah, they're banning it.
Yeah, well, yeah. I mean, Roundup actually binds to a very specific enzyme that's found in bacteria. So I think. Well, no, actually not in bacteria. It's the enzyme actually detoxifies Roundup. So I had that backwards. So basically, that's why the controversy with GMOs. And I'm not sure which plant enzyme that Roundup binds to, but rotenone is basically a small molecule that binds to, I think, something in the electron transport chain. So kind of like cyanide. So really, really nasty stuff.
Yeah, I mean, you'd have to consume a lot of Roundup yourself if you wanted to get an LD50 and then die from it. But yeah, right now, I mean, I know this because I do gardening and it's nasty stuff. It's powerful, but it's nasty stuff.
Yeah, it basically can give you Parkinsonian-like symptoms and all kinds of neurological problems. So, so yeah, toxic. So really hits mitochondria really well, and does bad things. So FCCP is a kind of, I think it's a cyanide-based compound. Anyway, also hits like either a cytochrome or something in the electron transport chain. So anything hitting things in the electron transport chain of your mitochondria is pretty toxic stuff. So you basically don't want to slow down or shut down ATP production in your body. That's kind of getting at the heart of everything. So what happens when you add these toxins? Well, it's lethal at a certain concentration, so percent alive. So they do this kind of lethality assay. But when you have the presence of this benzocumarin, it rescues them. So basically, higher percentage in the blue of these worms are alive when they're subjected to FCCP and rotenone. So suggesting that the mitochondria that were, again, this is their speculation here, but based on what they showed previously, that these mitochondria that basically got damaged by these compounds were turned over and new mitochondria were able to take their place, because you have increased mitophagy and everything else that's associated with it to regenerate mitochondria. So it's kind of more of the same on this. So they've got some really cool and funky assays here in this paper. So let's see, Mito-Rosella. So they have various stains that are active. So they have mitochondria that basically are, have been genetically modified to produce these pH sensitive reporter genes that basically, if the mitochondria fuse with lysosomes, it quenches the GFP levels because the pH is lower in lysosomes, more acidic. So they have this promoter that's neuron specific. TOM20, I believe, is a tag that targets things to I think the outer membrane of mitochondria. I think TOM stands for translocator of outer membrane, something like that. There's a muscle-specific promoter, and basically, they say this is statistically significant. So there's a lot of worms here. So it's kind of messy, but looking at these, I wouldn't have expected the p-value to be so low. But okay, there it is. There's the bar graph. There's the worms. There's your p-value. So I'm just presenting that to you. So supposedly you have more fusion of mitochondria with lysosomes, so more mitophagy happening when you add MYC. I don't know what concentration, no doubt within this range of, it's in the micromolar range, I believe. And kind of the same thing happening here. It's another reporter assay that they use. pH sensitive fusion, and they have another protein here that basically, so if you look at DMSO and MYC, so low and high exposure, it's basically just the exposure of this Western blot, I believe. MFP is, so this is a portion of this protein that they use that essentially, when it has a proteolytic site. So when it fuses with the lysosomes, it gets degraded, and you have this small bit that's basically broken down. So this arrow here is pointing to the bit that basically came off of this fused DFP protein that's fluorescent when it gets targeted to lysosomes. And the levels go up when you add in MYC. So essentially, you have all of this kind of evidence using these really, cool reporter strains that you have fusion of mitochondria with lysosomes, and that goes up when you have this MYC compound. And then over here, what they're showing is that if you knock down levels of DAF-12 using RNAi, I think DCT1 was implicated in mitochondrial fission. if you knock down DAF-12 or DCT1, which is again involved in mitophagy, that the percent alive when you expose them to rotenone goes back down, and also you have less cleavage of GFP. So essentially implicating DAF-12 in this process, which is the ortholog of the mammalian nuclear hormone receptor in C. elegans. This here is an assay that I've never performed before. It's based on a machine. It has a funny name called Seahorse assay because that's the company that invented it. So essentially, it's this device and this screen that looks for oxygen consumption rate in mitochondria. So if you have higher oxygen consumption rates, then the mitochondria are healthier. You have more electron transport chain, activity happening. So if you add in MYC, I think M is maximal oxygen consumption rate. But if you knock down DAF-12 and DCT1, those levels go back down again, implicating these also in mitochondrial health. So if you throw in your benzocumarin and you have downstream components, then not surprisingly, it blocks the activity of MYC, suggesting that at least for mitochondrial health, you need these downstream components for MYC to function. That's not to say that these benzocumarins might not have other beneficial effects in other pathways that are unconnected to mitophagy, but at least it seems this is the way that they're acting in mitophagy. And like I said, I'm still a little bit confused exactly how this benzocumarin is interacting with DAF-12, whether or not it is because my understanding is ligand binding to DAF-12 is a good thing, and it's going to switch on the transcription of TFAB or HLH30. But the data later suggests that if you don't have the ligand binding to DAF-12, and MYC, of course, inhibits that binding, then that is what's important for mitophagy to happen and lifespan extension, which we'll see in a moment happen. So I'm still a little bit fuzzy as to whether or not this benzocumarin is negatively or positively regulating DAF-12. So I'm a little embarrassed to say that. It's very fundamental, but I'm looking at this figure and I'm looking at the results of these assays and I can't figure it out. FCCP is an uncoupler. Okay, yeah. And glycosylate cycle is targeted by Roundup. Okay. So that was figure 3. Oops. And figure 4 is lifespan assays. So, it's sort of the heart of what we always want to look at when we look at whether or not something actually works in kind of promoting the health of an organism, especially when it comes to aging. Now, of course, they haven't looked at aging per se in this paper. They're looking at mitophagy and they're looking at mitochondrial health. But of course, increased mitochondrial health is implicated in aging. I believe it's one of the hallmarks of aging. A decrease in that. So you would expect, we would expect these benzocumarins to be effective in aged organisms. That being said, if the levels of of your nuclear hormone receptor are disrupted, then then that's going to be a problem for this benzocumarin to work. Excuse me. So, what's the take-home message here? Well, if you add in your mitophagy-inducing compound at a particular concentration, the worms live longer. Again, I don't know exactly what genetic background these wild type. I guess N2 is the genetic background. I'm not a worm person, so I don't know what that if it's typical to just use one strain background, or we should be looking at other strain backgrounds. So there's a percentage.
I can comment
Yeah.
on that.
Oh, yes, please.
Because I'm a worm person.
Perfect.
Yeah,
so you take over.
N2 is the wild type for C. elegans, and it's used by all the labs around the world. And typically, yes, we use just N2 as a wild type. Now, for example, the CITP, which is the C. elegans version of the interventions testing program, they do look at 3 strains of C. elegans. But 99.99% of researchers use this N2 as wild type.
Well, there you have it. Succinct and to the point. Thank you, Sven. So 99.99% of your researchers will use N2, but some people like to go the extra step and use 3 different strains. So here is the N2. So somewhere in the paper, they'll tell you how many worms there are. More than 1, less than 10,000. So how about that? So somewhere where we'll find that number in the materials and methods. So you get an extension. And then we'll just kind of zip through here, because what we can see is that if we knock out DAF-12 or HLH30, or we have lysosomal dysfunction, these are genes that are required for lysosomal activity, or or that are important for they're either, I believe, proton transporters, or basically important for lysosomes. DCT1 was involved, I believe, in the fission of mitochondria. So all of these things, if you knock these things out, which we saw earlier, blocked mitophagy effects, also block the lifespan extending effects of MYC. So that's a very nice tie-in there. Complex 1 dysfunction. Interestingly, they kind of mentioned this briefly. Complex 2, that's in the electron transport chain. I don't know what the relevance is here. They kind of mentioned it briefly. I think they were a little confused too, that MYC seems to have worse effect on these MEV1 mutants. And I'm not really sure what the, why there's this added stress if you have this particular mutant for complex 2 dysfunction that you don't see anywhere else. So I'm not gonna kind of belabor this point too much. Just keep that in mind. Might be important, might not be. Okay, so interestingly now they jump into a different assay where they overexpress human neurotoxic proteins. So amyloid beta 42, which is implicated in Alzheimer's, alpha-synuclein, which is, I believe, it's a alpha helical protein that's involved in vesicle fusion and tends to accumulate, I believe, in Parkinson's disease. TDP43, I'm not sure what is TDP43 expression in neurons. So let's see what assays. So I just want to make sure that this is in. Let's see, are these in human cells, this assay? Just trying to, give me one moment here. I'm just trying to figure out which organism this is in here. Say 4J. Let's see. Oh, so these are, okay, cool. So these are transgenic worms that are expressing human neurotoxic proteins. And this tends to also, mess up the worms. So this is percent of the worms that are paralyzed, so I guess have problems moving around. I don't know how many worms they use here, but they seem to suggest that, higher concentrations of MYC will prevent paralysis in worms that are expressing amyloid beta 42 in muscles. I didn't know you could do that with worms, but that's pretty cool. And if you overexpress alpha-synuclein in muscles, I guess there's a phenotype called thrashing, and that also basically they thrash less, so they thrash more if you add in c-MYC. and also TDP-43, which is, let's see, what is that? That's human RNA binding protein TDP-43, which is associated with several neurodegenerative diseases, including ALS. And it seems to be that they get greater lifespan in these worms. So I don't know, Sven or somebody else, do you have any comments on this kind of system where they're expressing human, human proteins that are
yes,
it works?
it has been done many times before, so this is something that is being used. And the paralysis, indeed, as you say, when you express A beta in the muscle of the worm, it's typically not done in the neurons. It's kind of difficult. It exists, but there are so few neurons, they are difficult to visualize, especially. So that makes why people don't do it, because if you do it in muscles, you can also put the worms under the fluorobino and actually see the protein aggregates in the muscle and quantify how much of protein aggregates there are and things like that. So that's why they typically express it in the muscle and not in the neurons. And so then the worm's basically paralyzed, so they don't move anymore. And thrashing, that's basically when you put a worm in liquid, it starts to swim a little bit like sperm, that is something you're more familiar with. And that is what we call thrashing. And so you can just count how many thrashes there are. And it's a measure of movement. So, of course, again, if you express a protein that is toxic, is it also in the muscle, I guess, then the muscle will have a dysfunction and they will not be able to thrash as often and more per minute time.
Cool, very cool. I didn't know that. Yeah, I didn't know that you had these ready assays where you can take these proteins that are involved in human neurological, chronic diseases and put them into worms and then kind of test out compounds like this. So that's pretty cool. Do you know? Again, this is slightly off topic, but do you know if? This is an obvious question, and the answer probably is obviously yes. But do they use these systems here to screen for drugs that then later they test in animals as a potential human therapeutic for Alzheimer's, for example, or ALS?
I assume this has been done, but I'm not this familiar with that particular subfield. So I assume someone has done it in a screening type of manner.
Yeah, it just seems obvious to me, right? You got a human disease protein that induces a very obvious aberrant phenotype in survival, thrashing, paralysis in worms, where you can basically, obviously worms are well suited to do high throughput screening, and there you go, right? You've got a drug screen. But anyway, very interesting, very cool. So now we're on figure 5, which is they now switch from worms back to mammalian cells. So they used mammalian cells for their initial screen to find this benzocumarin compound. And then they do all of this in worms because it's pretty easy to do in worms. You've got a lot of genetic control there. Now they go back to mammalian cells, and they basically do very similar things to what they did in worms. They, if I could summarize this in kind of one sentence, yes, you have improved mitochondrial function and mitophagy in mammalian cells, just like you had in the worm. And the mammalian cells they use here primarily in all of these figures are these mouse C2-C12 muscle myoblast cells, which they use because they have high energy demands, evidently, and they rely a lot on mitochondrial function. So that probably, gives them, cleaner results, because these cells are very metabolically active and very dependent on mitochondria. But first, what they do is they do this assay, which is interesting because I was looking up what this assay was. So in figure 5A is not mouse C2-C12 cells, but what it is are it's a human. So this is a human FXR, so human, like a nuclear hormone receptor that's being expressed in, so I believe it's the FXR activity. I think it's a reporter. These are non-human cells. I'm just trying to figure out if these were mouse cells. So I was looking at this in the materials and methods, but I couldn't really find out which cells they are. So if you go to the materials and methods, it says human FXR activity assay determined according to the manufacturer's protocol. They don't really mention the manufacturer. Reporter cells that are engineered non-human mammalian cells that have been specifically designed to express human FXR. But that leaves a lot to be explained as to what the non-human FXR expressing cells are. So if you can find that out for me, I appreciate that. And the ligand for FXR here is they have this, I think it's a synthetic ligand, I'm not sure, this GW4064, which is kind of equivalent to the dacacroic acid that was binding to DAF-12. So in this case, FXR is the ortholog. So let me just double check what GW4064 is. Sounds like a synthetic ligand. I think it's a synthetic ligand. Yeah, it is. It's a potent synthetic ligand of FXR. So again, you have, if you add in various concentrations of your benzocumarin, you block FXR activity. So you basically get less of this reporter being expressed in whatever these non-human cells are. I don't know if that's proprietary information. That are using this human, FXR system. So again, this kind of ties in with the other assays that were shown where, yes, FXR DAF-12 is important for the benzocumarin to work. The benzocumarin seems to be blocking all sorts of native ligands and also synthetic ligands from binding to FXR. And by blocking that, it seems to block the transcriptional factor activity of FXR. But yet we know from other experiments here that that you need FXR to transcribe things. So again, I'm just like a little confused here. Well, maybe a lot confused as to whether or not the binding of the benzocumarin is acting in a negative or positive regulatory fashion through to FXR with regards to everything kind of downstream. Whether you need the benzocumarin to be bound to the FXR or not bound for it to have its beneficial activities downstream. So I'm not clear about that. I'm not exactly clear how that's functioning. Okay, so now to these mouse cells, which are these high energy demand C2, C12 mouse myoblast cells. So essentially they're looking at, similar to what they did with C. elegans. So there's orthologs here of all these genes that are basically involved in either mitophagy or the functionality of lysosomes. So they look at all of these and they mention what these genes are in the in the paper. Scramble through and just maybe find one example of one of them. Yeah, somewhere in there. So it'll take me a little bit too long to find out exactly what each of them are doing. Some of them are involved in I know proton transport. Other ones are involved in various aspects of lysosomal functionality. So basically, you have higher expression levels when you add this benzocumarin. Lyso tracker red staining. So let's see. So this is basically what do we have here? Full change relative to DMSO. So just like with the worm previously, increased, red staining. This is a stain that stains active lysosomes. So it's slight increase here when they add this benzocumarin. BAF, I believe, is a negative control that they use to knock down the functionality of lysosomes. I don't know if they have a much stronger positive control than MIC, but I guess MIC is the best they have. So, similar to what we saw with the worms and the staining, the staining of lysosomes, this goes up higher. So you have higher, more lysosomes, more lysosomal activity taking place. So it's I believe, a pH sensitive stain for lysosomes. Let's see, what is DQ-BSA staining? So these are all basically indicators of increased lysosomal activity. There's a FACS sort here, so a flow plot for reactive oxygen species. So mitosox, I believe, is a stain for free radicals. So basically, so reactive oxygen species. Let's see. Oh, and G is the quantification of F. So rhodonone should have much more reactive oxygen species building up if you're blocking electron transport. DMSO as a control. And then MIC is obviously should kind of abrogate that. UA, let's see, what is UA? I think that's urolithin A. I think that's also a control that they use here. So MIC is going to basically, improve so essentially what you're seeing here is improved oxygen consumption rate, same stuff that we just covered in the last figure before the last figure. In these cells, you essentially have less reactive oxygen species. When you add in MIC, let's see, urolithin A, how is that functioning? That's, I think that's urolithin A. Let's see, figure G. I know we covered urolithin A in a previous paper. I just can't remember right now what its role is towards mitochondrial health. Let's see. UA was used as a positive control. Okay, well, that's good. Okay, we'll just keep it there as a positive control for now. Okay. And of course, cell viability was increased significantly when in the presence of rhodonone, just like we saw with with our C. elegans in the previous paper. So this is translating basically to mouse cells. So you're essentially seeing improved mitochondrial health across at least 2 different species, mice, at least in tissue culture, right? They haven't done this in a mouse, in vivo in a mouse, but in tissue culture, and also in vivo in C. elegans. So it's I like these health and anti-aging papers that look at small molecules that work kind of across the evolutionary spectrum, because that is really suggestive that these molecules have a lot of promise because they target some really fundamental mechanisms. And if you get improved health throughout all of these different species that are widely separated evolutionarily, I think that kind of bodes well as using these molecules as sort of a platform, maybe to make even better derivatives that can target really fundamental mechanisms that are very well conserved evolutionarily. That's my take on it. So we've had other papers in the past that have kind of done a similar approach. Yeah. So thoughts, comments, anybody? I'm still a little bit perplexed exactly how MIC is modulating the function of DAF-12 and nuclear hormone receptors with regards to its ligand, its native ligand. So I'm still a little bit fuzzy on that, based on how the assays worked and based on how the in vitro data, what the in vitro data has been showing. But you can't argue, at least with the end result, which is improved mitochondrial function and longer-lived animals. So.
Well, next question: is it going to work in humans?
It should, right? I mean, coumarins are this class of compounds that are known to have beneficial health effects, right? So I think, so yeah, it should work in humans.
Yeah. Yes, is the word you're looking for. Yes.
How well, we're not sure yet.
Could it be more research needed?
Yeah, more human research.
Human research needed is the groundwork, of course. And then you get people going, "Wow, no, it's worms, cheese," but they don't really seem to appreciate that conserved pathways like this may also be human relevant. But you tell them every time, and they'll keep trying out the same old criticism. What can you do? What can you do, Oliver? You can't do much. So there you go. So speaking of human trials, there are a few things entering or currently in trials now. So perhaps in a future journal club, it might be nice to, rather than just focus on a single paper, give a whistle stop to what's going on.
What did you say? You said focus on
the things that are currently in human trials, because they're actually not an insignificant amount of things. We've also had a recent update to the Pearl project as well, the rapamycin thing that we did. You can find an update on the site, but there is more coming very soon. So that's exciting. Quite enthusiastic about the potential of rapamycin. And but it might be nice to actually, perhaps in the new year, take a look, a recap of just what is in trials at the moment. And if you can't wait, won't wait, visit our website and check out the Rejuvenation Roadmap, which is a visual database showing you exactly what clinical phase things are in. And there's some that are in 3 and even sort of extended phase 4s, and some that are about to. So I don't know. I'm just putting that up the flagpole there, Oliver, to see what you think. Running that up the old pole, see if you'll salute it. Really lazy salute. Sorry about that, guys. It's straight up, straight down, Navy style for me. Oliver, I don't know how you guys do it. You do it?
I call that the Benny Hill salute.
That's very British Army, sort of 1940s. So very amusing. I still think they salute like that. Not quite so exaggerated, but there you go.
It was just a thought occurred to me while I was reading this paper, because again, these benzocoumarins are very, at least this paper here suggests that you need this nuclear hormone receptor to be present. You need this TFEB protein to be present, and I believe they mention in this paper that TFEB, the transcription factor that is upregulated by these benzocoumarins, goes down with age. And if that's the case, then it would be interesting. I don't know if anybody's kind of done these types of experiments with humans. If we know, if we have a hypothesis that some of these compounds require a certain level of the downstream whatever is being regulated downstream, then obviously having lower levels of that would make these compounds less efficacious for those individuals. So it would be interesting to, if you're going to do a human trial like this, to kind of get baseline levels of the expression of these downstream, factors, and see whether or not the improved health benefits correlate with having sufficient, levels of these factors that are being targeted. Sort of like the reverse of of cancer drugs, right? So if a cancer drug targets a particular particular receptor that's overexpressed, then obviously it's going to be not efficacious if your patient doesn't have an overexpressed receptor, right, to be targeted, because there's a bunch of different mutations that can happen in the same pathway. So you want to diagnose and make sure that the patient is a suitable candidate for that drug, which is targeting what's being aberrant. So maybe in this case, if you're going to be giving longevity drugs to individuals, you might want to make sure that that whatever the factor is that this drug is binding to is still at a sufficiently high level. If it is, if the levels do go down with age, that you introduce these compounds early on, or maybe at a stage where they can be efficacious for an individual. And if they don't work, then you can see if you can figure out whether or not it's not working because those particular participants are lacking that downstream component. So I don't know if anybody's kind of looking into that when they're doing these kind of drug molecule studies with humans, or if it's still a little bit vague on the activity. But at least that was, I believe, mentioned in this paper that that TFAB has been studied. Yes, age-related declines in TFAB activity or expression disrupt ALP. So there are age-related declines. So yeah. Just kind of tossing that out there as a thought that maybe we should look a little bit further and see if we can figure out if we can target individuals that would be most benefited by a particular small molecule. And if it's not working, probably for those reasons it might not be working, and there might be other molecules that can play a role. So that being said, I'm out of thoughts right now for this topic.
But hopefully you're not completely out of thought. Problem. Maybe we should give you some feedback.
Yeah, maybe. I'll take a look and see if I can
get those mitochondria supercharged. So yeah. So interesting stuff. Mitochondria continues to be a fascinating topic. See, I'm following things like the Mitosense project over at Sense Research Foundation, and I've definitely encountered Tom before, you mentioned Tom, and TFAB. I'm pretty sure I wrote about TFAB research about 3 weeks ago. People were looking at it again in the context of longevity. I'll have to go back and double check, but TFAB sounds very, very clear. And I think it was possibly a potential target that George Church. I need to go back and check around. I think that was a possible candidate for gene therapy targeting. Church lab that was talking about it. This has got to be about 4, maybe 5 years ago. So comes round in circles, though.
Oh, Micah, I've been told that your mic. Well, we're probably wrapping up right now, pretty soon. But I think your mic was a little bit crackly.
Yeah, it's knackered. I've got to get a new one.
Okay.
I'm too hard on them, guys. I'm too hard on them. Okay. It's a gaming headset. I throw it around. I need a new one.
Okay. So with that, I wish everybody happy holidays. 10 more. What is it? 10 more days. 100 more days. What month are we on? We're in December, right? So yeah.
100 more days.
Yeah. Happy Easter, everybody.
Something's gone very wrong with the Earth's orbit if it's another 100 days.
Happy St. Patrick's Day. No. So Merry Christmas. Happy Kwanzaa.
Oh yeah, I was going to say
Happy
Kwanzaa.
Hanukkah, everybody. Whatever your celebrations are. If you have a shrine to Baphomet in your house, whatever it is.
We can't do that, Oliver.
Okay. Okay, so with that, cheers, everybody. I'll be taking off, and Micah, I'll send you a copy of the paper.
Thank you very much.
All right, take care, everybody.
Bye-bye.